Yes, sir.
Okay, thank you, Rana. We are starting a lecture on nanomaterials, and nanomaterials, they are in the size of nanometer. The particle within the dimension size of nanometer is called nanoparticles. And nanoparticles is the particle size is 1/billion of a meter. That means 1/billionth of a meter. And what does it mean? Let us see things in the nature. In the nature, an ant is 5 millimeter in size approximately, and a dust mite is 200 micrometer, and fly ash 10 to 20 micrometer, and red blood cell, our red blood cell in
That's it.
our body, or white blood cell, 2 to 5 micrometer. And up to this, anything we can visualize by using light microscope. But if you consider the ATP synthase, which is an enzyme involved in synthesis of ATP, its size is 10 nanometer in diameter. 10 nanometer in diameter, you cannot see it in the under the light microscope. We need to see it under electron microscope. Similarly, DNA molecule, you see, DNA molecule is around 2 to 3 nanometer in diameter, and the DNA molecule is also within the size of the nanomaterial. In our earlier class, previous class, we discussed about the size dimension of a nanomaterial or nanoparticle. Nanomaterial or nanoparticle within the size dimension of 1 to 100 nanometer.
Nanometers.
So the study of uh, particles within the size dimension of 1 to 100 nanometer is called nanotechnology. And
Technology.
nanotechnology is an emerging technology in the field of electronics, in the field of biology also, and it is becoming a powerful technology in agriculture. So in our previous lecture, I discussed about the why nanomaterials are different from in properties, physical and chemical properties, than the molecules. So molecules in the when they are aggregated to form the structure, crystal structure or particle structure within the range of 1 to 100 nanometers, they behave differently. Their properties dramatically being changed, and these new properties are important for their application in the practical life. I told you in electronic field, nanomaterials have been used in, electronic chips or IC or in many devices, electronic devices, and they can be used to store the data. And I told you in previous lecture that DNA molecule, scientists did lots of experiment to use DNA molecule as it is a nano size to store the data. A few milligram of DNA molecule can store the data available all over the world. So you can understand things man-made. I discussed here all our natural things from millimeter to nanometer. Here you can see things man-made. Man-made, for example, head of a pin, 1 to 2 millimeter,
Millimeter.
and electron or electrical device in your computer or other mobile phone. All the devices are in nano size. So their size is, you see, micrometer in size. They are micro. So micrometer means light microscope, you can see them. And then you can see nanometer size, outer ring spacing, on the particle nanometer, we can use even in the CD to store the data nano
Data.
particles. So self-assembled materials, nano electrode in nano size, for example, carbon containing buckyball structure or fullerene structure or carbon nanotubes, they are used in nano electrode to make the nano electrode. Similarly, quantum dots electronics are different types of materials which are useful in making the smart electronics chips or materials. They are nanometer in size, and they possess very strange properties, and these strange properties are the basis of their practical use. So nanometer scale, 1000 nanometer equal to 1 micrometer. So virus and DNA are natural nanomaterial. If I ask you a question, can you cite an example of natural nanomaterial? Then you can say virus and DNA. Coronavirus, we are now under the amid of coronavirus pandemic caused by the severe acute respiratory syndrome coronavirus 2 SARS-CoV-2 virus. SARS-CoV-2 virus is also a nano particle. All the viruses are nano material.
Material.
So SARS-CoV-2 virus is around 60 to 65 to 120 nanometer or like that. So the DNA molecules under transmission electron microscope, you can see this double helical structure like that. And this is the microscopic image and microscopic image of the nano materials like coronavirus crown-like structure. Every day in the television you can see it. It is a nanomaterial. So application as microchips in the computer nano materials have been used for a long time, and lots of electronics, we are working now in the digital world. So all of we are using the nanomaterials. And nanomaterials application also available in other area like, our practical life, sunscreens and cosmetics. Many sunscreens and cosmetics contain nano particles, and those nanoparticles protect us from the UV light when you go outside. That means exposed to the sunlight. Sunlight, contain UV, which is damaging for our skin. So people use sunscreen and cosmetics. They are the nanomaterials. So antibacterial effect of silver nanoparticles, people use in the shops so that they are antimicrobial, so fungus cannot grow, and this type of nanoparticles can be used in the cosmetics, and silver nanoparticles also used in the soap. Bath soap, we use, we need antimicrobial materials. For example, Lifebuoy soap, they advertise that it contains, silver antimicrobial silver nanoparticles. So you can understand now what the practical application of nanotechnology, not only in electronics, in our life science. So nanoscience, how can we define nanoscience? The study of structures and unique properties of matter at the nanoscale. So the study of structure and unique properties of matter at the nanoscale is defined as nanoscience. And then we can have, we can understand that an nanoscience is an interdisciplinary field of science combining physics, material science, chemistry, and related disciplines. That means nanoscience is an interdisciplinary field of science. It combines physics, It combines material science, it combines chemistry and other related disciplines, even biology. So derivative of cholesterol, you know, cholesterol in our body, sometimes when you check your lipid profile, total cholesterol is counted. Cholesterol structure is like that, and cholesterol is essential for our membrane and our cell, but a higher level of cholesterol is problematic for our cell. So first example of a liquid crystal is cholesterol. Cholesterol can be crystallized and can make nanoparticle, and it was studied in 1888. Unusual properties to melting points if it becomes the crystal. Unusual interactions with light, no interest at that time, but now it is interesting. So liquid crystals are a state of matter which has properties between those of conventional liquids and those of solid crystals. So liquid crystals, cholesterol behave very unusually, very interestingly, and it has been studied in late 19th century. What is nanotechnology? So we know now nanoscience. Now let us know what is nanotechnology. Nanotechnology is the understanding and control of matter at dimension of roughly 1 to 100 nanometers, where unique phenomena enable novel applications. Unique phenomena, that means any molecules, when you make them particles within the range of 1 to 100 nanometers, they possess unique properties, and unique properties are the basis of their application as nano technology. It encompassing nanoscale science, engineering, technology. Nanotechnology involves imaging, measuring, modeling, and manipulating matter at this length scale. So nanotechnology, if you go this website, you can get more information. This is a nice definition on of nanotechnology. So is nanotechnology new? In our previous lecture, I also told you that it is not really new branch of research or science. Other names used in the past: chemistry, biochemistry, microbiology, atomic molecular physics, molecular chemistry, or molecular engineering. It has been around centuries in the research. DNA double helical structure, and other virus, these are the virus particles, they are the nano materials and have been available in this earth for a long time. Nanoscience is concerned with the study of novel phenomena and properties of materials that occur at extremely small length scales. Extremely small length scale means length 1 to 100 nanometer. Nanotechnology is the application of nanoscale science, engineering, and technology to produce novel materials and devices. So you can now differentiate nanoscience and nanotechnology. So nanoscience is the study of novel phenomena of the material at nanoscale, but nanotechnology is the application of nanoscale science. The novel properties that we know through the nanoscience can be used as a technology, then we to prepare new materials or new device, new materials, sunscreen or other antimicrobial substances, or nano chips, electronic devices, or like that. So I hope it is clear to you. So nanoparticles. Nanoparticles, you may be asked to define nanoparticles. Nanoparticles are among the first nanoscale material to be directly useful in biology. Fluorescence particles labeled with antibodies as tags that do not photobleach, and supramagnetic magnetite particles coated with dextran as image, image enhancement agent in the magnetic resonance. We can use in the fluorescence imaging microscopy, this type of nanomaterials to visualize a cell or cell organelle or a molecule in the cell. It is very important in the cell biological study. Eventually, these small particles must be made much more useful and informative if they are to play an important role in understanding the working of the cell. So how cell works, if you want to visualize mitochondria, if you want to visualize nucleus, if you want to visualize chromosome or a particular protein in the cell, you can use fluorescence tagging. That means nanomaterials that can bind and emit the fluorescence. You can see the cellular activity under confocal laser scanning microscope. And in previous class, I told you even gold at nanoscale you can dissolve in water and can, if you pass through the light, different size of the gold particle emit different fluorescence light, and these lights can be used in the industrial processes. Even MRI, magnetic resonance imaging in medical science, very important technology. Nanoparticle can be used to visualize cancer and other problems in the cell or tumor. For example, if you use the dye, nano dye, and then MRI can visualize it. So in medical science, nanotechnology has been used for a long time. Even drug delivery, specific drug can be delivered to the specific object of the pathogen like cancer cell directly without harming the other cell. Carbon nanotube, I told you, ISIMA in 1991, not very old time, they prepared in the laboratory this type of carbon nanotube or carbon nanosheet, and they are very, strong and only carbon containing compounds. And you can put the molecules inside the nanotube and can use in the medical, as well as you can make polymeric substances, very hard material. You can prepare synthetic materials by using these nano materials. In our previous class, we discussed the diamond and coal. They are both composed of carbon, but diamond you see glittering and carbon is black due to the arrangement. Carbon in the coal arranged like this way, but in a diamond, carbon arranged in that way. A diamond is the hardest material and very glittering, and it is due to the nanotechnology. That means naturally nanotechnology, diamond is an example, and coal is an example that non-nano particle arrangement is different. And if you see 1 kilogram of coal in the market price, and if you consider the diamond is the most expensive jewelry material, and it is due to the their carbon arrangement and structure. So fullerenes, I told you, fullerenes are a recently discovered form of carbon by Proto, Carl, and Smalley. Please visit Wikipedia and see these three Nobel laureates and how they discovered the Buckminster fuller, that is this type of fullerene structure, ball-like structure. Inside is hollow, so in 60 carbon containing typical football-like structure. So inside you can put any materials. I told you in previous class, for example, if you want to prepare urea, nano urea, urea molecules, if you place inside this fullerene structure, buckyball structure, then they will release slowly. Even you can make nanoparticles which release very slowly, so you can reduce the wastage of urea through volatilization, as well as leaching or washout. and you can increase the efficiency of fertilizer dramatically. Not only urea, other fertilizers can be prepared like this way. So application includes this type of structure in cosmetics, pharmaceuticals, lubricant for engine parts and electronics, as well as fullerenes can be used in agriculture for making the some uh, fertilizer, slow releasing fertilizer. For example, anti-aging facial cream available in the market. They are also use this type of carbon structure, and they have the carry the compounds, anti-aging compounds. So I told you the cadmium silicon single crystal is also nanoparticle, and gold, when they are nanoparticle, they can be dissolved and pass through. You can dissolve the gold in which is impossible to dissolve even in a single acid. I discussed this one: colloidal nanocrystal synthesized by chemical means, 2 to 6 nanometer. They are like this, and strong blue shift of photoluminescence emission. So luminescence light, photoluminescence light, you can make a bulb, light bulb, different colored bulb you can make by using nano materials in the surface of the bulb. Surface passivation by the organic molecules, so core shell structure yield high luminescence efficiency. So now electricity consumption can dramatically be used by using different luminescence efficiency of nanoparticles, so light emission capacity can be increased. In our earlier lecture, we discussed how can we prepare nanomaterials. One method is the top-down method. That means macroscopic, visible, big matter. You can break down them and make the nanoparticle, as well as molecules. By aggregating them in chemical reaction, you can make crystal structure within the size dimension of 1 to 100 nanometers. So if you are asked a question that how can we prepare nano structures or nanomaterials, there are two possible ways. One is top-down approach, materials available in nature. You can break down them and up to the nanometer size, and then there will be the nanometer nanoparticle, and molecules. Molecules are usually 10 to the power minus 10 or like this, very small, and even you cannot see by using the small molecules, you cannot see by using transmission electron microscope, but big molecules you can see, like DNA molecules or ATPs. Enzyme you can see by electron microscope, but molecules usually we cannot see small molecules like antibiotics. So aggregating of the molecules, you can make the crystal structure, and if structure ranged from 1 to 100 nanometer, then they are the nanoparticles. And those nanoparticles, depending on the size of the particle, the properties obviously varies. So assembling nanomaterials bottom up or top down, these are two methods. So DNA molecules is self-assembly of nucleotide uh, uh, make the DNA molecule in the biology. So assembly taken place in the nature, and you can do assembly in the chemical reaction. So by using different chemical reaction, you can make the nanoparticles, and nanoparticles can be visualized by the use of electron beams, or by the use of electron microscopy. By using electron beams, light, or lithography, you can break down the materials and make the nanomaterials. So atomic force microscopy can visualize the particle size of the nanomaterials. So these are very important. For example, Mercedes-Benz car, or Mercedes-Benz car, the surface paint is not the normal. They are the nanoparticles. This is why they can they are highly resistant in weather and not easily, degrade and not easily fade. And these are very useful technology. So top-down miniaturization photolithography is very important technology to make bigger molecule into miniature size, that is nano size, and it is called photolithography from Latin word light stone writing. Photolithography is the process of transferring geometric shapes on a mask to the surface of a silicon wafer. The steps involved in the photolithographic process are wafer cleaning, barrier layer formation, photoresist application, soft baking, mask alignment, exposure and development, hard baking. So this is a systematic process of making this type of nanomaterials. So these are the very advanced technology for making nanomaterials by using photolithography. And very interesting process. And molecular bottom-up strategy for molecules to the nanoparticles. So energy interaction, information. So they work together, selective binding of substrate by a receptor. So the molecular recognition. For example, a receptor. Receptor is a protein present in the cell membrane, and agonist, a molecule can bind to it and make the assembly. So these are naturally happening. But in artificially, you can do the molecular assembly by chemical reaction to make the nanomaterials. For effective molecular recognition, receptor and substrate should exhibit steric complementarily shape and size, interactional complementarity, large contact area, multiple interaction sites, strong overall binding. For example, we use antibiotic. Some antibiotics are very effective, some are less effective. We use pesticide. Some are very powerful, some are less powerful. For example, fungicide, we use strobilurin because all these drug molecules, they have the receptor protein in the cell membrane of the target organism. The more surface area, large contact area of the drug molecule and the receptor, they function higher, stronger. So stronger binding is very important, and it is associated with molecular recognition. So it is also a natural nanotechnology. Self-assembly of nanostructure, you can see this is a nucleotide. This is a nucleotide. In DNA, lots of hundreds of nucleotide unit, join together and makes this DNA double helical structure. This is a self-assembly of nanostructure. So self-assembly of nanostructure taken place in the nature, and from the nature we learn it, and now you can do it in the laboratory to make new nanomaterials. So in the intersection of nanotechnology and biology, nanotechnology offers biology new tools. Biology offers nanotechnology access to new types of functional nanosystem, because in biology, for example, this is an ATPase enzyme. If you know the detailed structure of this one, then you can synthesize this type of molecules in nature, and even you can make the energy currency, that is ATP, in the synthetic biology or system biology. So natural assembly of materials or nanotechnology existing in the biology, giving us lots of new information for making synthetic or artificial nanomaterials. So you can make molecular machines, molecular robot based on this knowledge. Molecular organelles, small organelles in the cell, lots of organelles, for example, mitochondria, chloroplast, all are, nanostructure. So in future, people will make this type of compounds. So the ultimate in functional nanosystems, biological nanomachines, people or scientists can be able to do that. populate the cell, the ribosome, sodium potassium ATPase. This is an enzyme. Flagellar micromotor of bacteria. Bacteria has a flagella and there is a motor system at the base of the flagella. Linear micromotors of muscles. Muscle has linear micromotors and of microtubules, that means cell cytoskeleton, that organize and move to the cell, voltage gated ion channel, DNA replication complexes, multimeric membrane receptors, and photosynthetic reaction center like photosystem 1, photosystem 2. All these are, natural nanoparticles. And if you know all these, molecular machines, it would help us astonishingly. They are complex and they will help us to construct and design, a molecular machine in future. So nanotechnology in future in biology is becoming a robust area for research. Atomic force microscope is used to visualize the nanostructure. Because normal microscopy you cannot study. Nanomaterials, their properties are not only varies depending on their size, but shape is also very crucial. So their size and shape both, in a complex way, they show the properties, and those properties are useful. So this is the last, slide for today's lecture. Selected categories of nanotechnology applied to food and agriculture. Now the question is, in food and agriculture, where we can use nano technology. In agriculture, bioenergy or products, single molecule detection to determine enzyme and substrate interaction, cellulases in production of ethanol. For example, bioethanol can be produced by using the biomass. How cellulase enzyme, work on the cellulose to make the ethanol. If you know that, then you can increase the productivity of bioenergy. So materials from biomass. Biomass means the matter we get from the living organism. For example, straw, sugarcane bagasse, all dry matter we get from the living organism, even human or plant, they are called biomass. So biomass consists of carbon, hydrogen, and nitrogen. And if biomass can be used to make the bioenergy like ethanol. So how different enzyme works on biomass and to make the energy like ethanol or butanol useful for running your car or engine, you can understand the role of cellulase. Cellulase is a polymer, a big molecule of amino acid that is enzyme, and it is a nanoparticle. Agrochemical delivery of pesticide. The pesticide like insecticide, fungicide, nematicide we are using in the modern agriculture. 99% of them are, absorbed by the environment. Only 1% are practically used by the target organism. This is why our efficiency is very poor. So if you use nanotechnology, you can increase the efficiency and delivery technique of the pesticide to the target. Similarly, fertilizer also, other agrochemicals more efficiently only when needed or for better absorption. Animal production, delivery of growth hormone in a controlled fashion, then you can accelerate the growth of the animal. Identity preservation and tracking by using nanosensor. For every animal you can use, or every plant you can use nanobiosensor, and by using your computer you can understand the requirement of that animal, if there is a disease or if there is a fever, or if plant is affected by the insect, or if irrigation is needed. So all the things you can monitor by using your computer, by using nano sensor or chip. And this will lead to the precision agriculture, more efficient. Animal for plant health, direct animal pathogen such as foot and mouth disease, virus, detect plant pathogen very easily. Nanomaterials, some of the materials can bind with the surface receptor of the pathogen, and then they illuminate, and they show the fluorescence light, and you can understand very easily diagnose the pathogen in the plant as well as in the animal, and even cancer can be detected in the blood by using nano detector diagnostic tool. Animal medicine, deliver animal medicine to the right place. So drug delivery is very, very important area, is becoming a big industry because then you can deliver the drug in the infected area. For example, if you have infection in your finger, we take the antibiotic, whole body is toxified. No meaning to create the stress to the healthy cells. So if you direct the drug to the affected cells or the pathogen, then it will be more precise. You need more less toxin, and you can keep other cells free from the toxin. So plant production, delivery of DNA to plant towards certain tissues, targeted genetic engineering. Nanomaterials are now people are using for gene delivery or genetic engineering. And some of the nanomaterials have been found to trigger the enzyme activity of photosynthesis. As a result, faster growth can be possible. Sensing, I told you, detect chemicals or foodborne pathogens, biodegradable sensors for temperature, moisture by using the sensor. Even the presence of heavy metal or other toxins in the environment, by using biosensor, you can do, nanosensor, you can do it. Safety, selectively bind and remove chemicals or pathogens from the environment. So safety can be enhanced. Packaging, this is a very important. I told you in earlier lecture. For example, horticultural product, most of them are climatic and they become, perishable very quickly, and due to the release of ethylene gas. But if you make a package container which quickly absorb the ethylene, and then autocatalytic properties of the ethylene will be reduced, then you can increase the shelf life of the vegetables and fruit, as well as if antimicrobial coating on the packaging are there, then they stop the infection by the microbial organisms, pathogens. As a result, you can increase the shelf life. Healthy food, better availability and dispersion of nutrients, nutraceuticals, and additives, can ensure our healthy lives. So this is all for today's lecture. Now I would like to discuss with you something more, and you can ask me some question. Now it is your turn to ask some question. How did you understand, and what is your impression about the nanotechnology, and how do you feel? Because this is obviously a new course for you, and you shall learn lots of new topics, new keywords, new terms. So tell me if you have any question, or what is your impression. Did you really understood
Nanotechnology,
the lecture?
yes, sir. Nanotechnology is very interesting subject to me.
I cannot hear you. Can you increase the volume of your? Yes, please continue.
Sir, I understood your language, your lecture. Nanotechnology is very interesting subject, I suppose.
So my question is, in horticulture, as a horticulturist, where do you think nanotechnology could be used in a best way, and we can make more profit in agriculture.
So, fruit spoilage, fruit and vegetable spoilage.
By using nanomaterials in the packaging, you can reduce the spoilage of the fruits and vegetables. Very good question. Any other area you feel now that in horticulture we can use nanotechnology?
Or insect pest,
Yes,
insect.
because horticultural products are also prone to, different insects and pests. So nano pesticide can be helpful. Do you think nano fertilizer can be also helpful for horticultural productivity?
Yes, sir. Nano fertilizer is
Yes,
also useful.
because horticultural crops, you need to apply the fertilizer also for accelerating the growth. So for the growth promotion and nutrition, better nutrition, you can use nano fertilizer. And any other, even in the processing and packaging, if you process horticultural products, for example, fruits and vegetables, and if you make the cane with nano film inside, you can preserve for longer time any perishable uh, horticultural crops, vegetables, fruits. Uh, do you
Sir,
have any other question?
transportation also be easy.
Transportation.
Yes, sir.
Yes, if you use nanomaterials, then you can transport the material, horticulture product for longer distance, and it would facilitate the marketing, and farmers will get the benefit because if shelf life is increased, for example, mango in our country, when you want to export the mango to other countries, in that case, mango is ripened very quickly, and then due to production of ethanol uh, and autocatalytic behavior of ethanol, mango ripen very fast and then become rotten. In that case, if you use nano packaging and container, uh, and if they absorb the ethanol, then autocatalytic properties of the ethanol will be reduced, and mango can be preserved for longer time. And another point is, I forgot to mention, you can make nano film on the surface of the fruit. For example, in Europe, apples are coated with wax nanofilm. So in that case, a loss of water from the fruit also not possible or dramatically reduced, and pathogen cannot invade the fruits, which ultimately enhance the shelf life of the fruits, and it can be used in the if safer nanomaterials, polymeric nanomaterials, we can use natural polymeric nanomaterials like nanomaterials from the cellulose and other natural substances. For example, chitosan or chitin. Then we can make biofilm, nanofilm on the surface of vegetables and fruits, and you can enhance the shelf life dramatically, which increase the profit of the farmers, because the market channel will be extended to longer, even in the from one continent to another continent, and farmers would ultimately get the benefit, and we can dramatically reduce the loss of vegetables and fruits, because naturally nearly 30 to 40% of the climatic fruits and even vegetables are lost during the marketing channel, as well as in the retailers. So we can reduce that big loss, post-harvest losses, by using nanotechnology. Any other idea or question do you have in your mind?
No, sir.
So what is your impression? Is it interesting course to you?
Sir, interesting.
You are only one student, registered for this PhD level course, Nanobiotechnology. I am really impressed, but it depends on your strong enthusiasm. I shall try to train you, but it is important for you to read lots of literature through browsing the internet, Google. You can find lots of, new advancement in nanotechnology. Any question? What I shall do today, today's lecture, I shall, upload in my YouTube channel and share with you the link so you can visit there as many times as you like. You can listen and you can store it and uh, and if you have any question at next class, you can ask me question or you can ask question through email. Uh, Last chance, any question?
No, sir. No question.
No, sir. Thank you so much. And I hope to see you next week. Have a nice weekend to you.